The next presentation takes us to the field of microbiology. The presenters are Davey and Katie Barufka from Montgomery Blair High School in Silver Spring, Maryland, and Langley High School in McLean, Virginia.
Hi. First off, we'd like to thank the Siemens Foundation and College Board for having us here once again to present our research. It's truly an honor. So Leishmaniasis is a debilitating condition that currently affects 12 million people worldwide. It is caused by protozoan parasites that are spread by phlebotomous female sandflies, which causes around 60,000 deaths per year. This is becoming more and more of a global issue, for there are 2 million new cases reported each year. Our study focuses on cutaneous leishmaniasis, which clinically represents itself in chronic skin lesions. Cutaneous leishmaniasis is prevalent in parts of the Middle East, parts of Africa, Central America, and South America, and is caused by the parasite Leishmania mexicana in the Americas. It is becoming more and more of an issue for the U.S., for thousands of U.S. troops have gone over and fought in the Middle East and actually contracted these lesions. The picture we have up is actually of a U.S. troop who has a lesion on his face after coming back from war. Also, the presence in South America and Central America is also one of another issue, because it may start to creep up, and we have actually found phlebotomous female sandflies in Texas and New Mexico, which is what causes this disease. Now, there are no effective vaccines available for cutaneous leishmaniasis. Although there have been many attempts in the past of vaccination strategies, none of these have been fully effective. The oldest form of strategy is leishmanization, where the individual would actually intentionally inject themselves with a live virulent parasite. This is called leishmanization, but it was abandoned for it was unsafe. The individual would actually develop the chronic skin lesions all over their body, so this practice was abandoned. Kill whole cell parasites, subunit recombinant proteins, and DNA-based vaccines were safe. However, they did not provide long-term immunity, and they lacked efficacy. Subunit recombinant protein actually was backed up in studies by NIH how it lacked efficacy because it could not provide immunity when challenged with the sandfly, which is what would actually transfer this disease. So my partner and I, we thought that targeted deletion of a virulence-associated gene would be the most promising approach, for it's promising in other areas. It is safe, and it does not cause the overt disease. And to understand which gene we want to delete, we want to understand the life cycle of the parasite. So Leishmania mexicana, this parasite is alive in two forms. It is in the sandfly stage and the human stage, the promastigote and the amastigote. The promastigotes are larger and flagellated, and they exist in the gut within the sandfly. And they use all the sugars to get energy for glycolysis to occur. This is how they acquire all their energy. When the sandfly takes a blood meal from the uninfected host, the promastigotes are injected into the bloodstream and quickly phagocytized by macrophages. Inside the macrophages, the promastigotes differentiate into amastigotes, which is the infectious form of the parasite. The amastigotes are smaller and unflagellated. And inside the macrophage, they proliferate and essentially cause the macrophage to burst, spreading to other parts of the body. So if we want to break this cycle between stages 3 and 4, so that it actually infects the macrophage, so it can build up immunity, but it doesn't keep proliferating, so it won't burst, and spread to other parts of the body. So amastigotes are different from promastigotes in the sense that they don't have the sugar to rely on for glycolysis, so they're highly reliant on their mitochondrial functions for energy. So my partner and I, to find a certain target that's right for deletion, we wanted to focus on a gene that is highly associated with mitochondrial homeostasis. So endonuclease G is a mitochondrial protein. We were doing research, and we came across a paper on endo-G, and we came across a paper that was in a Nature article on cardiac hypertrophy, or enlargement of the heart. Now, this paper was talking about mice that had these enlarged hearts that lacked endo-G. They couldn't make a distinct connection in that specific article, but they saw that there was lipid accumulation in the mice, which led to a conclusion that there was impaired mitochondrial functions. So my partner and I thought, my partner and I hypothesized that if we delete endo-G in the protozoan parasite Leishmania mexicana, will it disrupt the mitochondrial functions and thus lead to the attenuated and reduced virulence of that parasite as well?
So the first step in testing our hypothesis was to remove endonuclease G from the Leishmania mexicana parasite. And we did this via homologous recombination. So figure A shows a gene locus encoding endonuclease G. The schematic depicts that endo-G is being replaced by two drug-resistant markers, NAT, or norceathrisin, and BSD, or blastocidin. Two drug markers are required because the Leishmania mexicana genome is diploid, meaning that it has two copies of every gene. After a series of PCR and cloning reactions, we generated a final gene replacement construct comprising of a 5' UTR, or untranslated region, and a 3' UTR separated by the drug-resistant marker. This final construct was then transfected into the Leishmania mexicana parasites, which were being grown under BSD and NAT drug pressure. Figure B shows a series of southern blot confirming the absence of endonuclease G. Figure C actually shows a western blot, also confirming the loss of endonuclease G. Anti-tubulin antibodies were used as a control to ensure that equal amounts of protein were loaded into each of the 3 lanes. So now that we generated a successful null mutant, we had to see whether or not any of the mitochondrial activities of the parasite were impaired. So we did a measurement of ATP, or adenosine triphosphate. As part of normal mitochondrial physiology and oxidative phosphorylation, ATP is generated. This figure shows 3 strains: the wild type, the endonuclease G knockout, and the knockout re-expressing endo-G, or the add-backs, for both promastogotes and exenic amastogotes. Exenic amastogotes are simply promastogotes that differentiate in culture into amastogote when the pH is decreased and temperatures are increased, replicating the conditions within a macrophage. So in this experiment, we found that for both the knockouts in promastogotes and exenic amastogotes, there's less than half the ATP concentration that was present in the wild type. Furthermore, when endonuclease G was re-expressed, the concentration of ATP was relatively restored. We also see that for each strain of the exenic amastogote, there's a greater concentration of ATP than the relative promastogote strain, confirming our initial idea that these amastogotes are far more energy dependent and depend more on their mitochondria than the promastogotes do. So we already see mitochondrial defects because of the removal of endonuclease G. The next thing we did was a measurement of reactive oxygen species, or ROS. As part of the electron transport chain and mitochondrial respiration, these toxic free radical oxygens are generated. Most normal, healthy cells and mitochondria have elaborate antioxidant capabilities that are aimed at neutralizing these toxic free radical oxygens. So in this experiment, fluorescence by our reagents was directly proportional to the concentration of reactive oxygen species. We see in this figure, for both promastogote and amastogote knockouts, there's almost 20 RFU, or relative fluorescence units, greater than both the wild type and the add-back. This reveals that there's a greater concentration of toxic species because endonuclease G has been removed, and the normal detoxification mechanisms of the mitochondria are now dysfunctional. Finally, we did the measurement of cell viability, or the determination of living and dead cells within a cell sample. Most normal cells maintain a reduced state in their mitochondria and cytosol. During the electron transport chain, there are many reducing metabolites, NAD, NADH, FADH, FADH2, and these all maintain the reduced state within the mitochondria. So in this experiment, fluorescence was directly proportional to the concentration of these reducing metabolites. For both promastogotes and amastogotes, we see in the knockouts that there's much lesser fluorescence, indicating that these knockout cells actually have lesser concentration of reducing metabolites. This means that the endonuclease G knockouts are not able to maintain a healthy, reduced state within their cytosol and mitochondria. And finally, we wanted to do an experiment that was unique to the Leishmania mexicana parasites. So we looked at the kinetoplast, a unique organelle to all trypanosomatid parasites, which includes Leishmania. The kinetoplast is highly associated with the mitochondria and is essentially the mitochondrial DNA. It's composed of minicircles and maxicircles, which are actually disentangled during replication. So we stain the nuclei and kinetoplasts of both wild type and knockout cells. What we found is that in the wild type, there was one nucleus and one kinetoplast for a majority of the cells, which was normal and expected. However, in our endonuclease G knockouts, we saw many cells that were aberrant and multinucleated. Nearly 10% of all the cells that had the knockout endo-G actually had more than 3 nuclei, while only 0.5% of all the cells that were wild type had greater than 3 nuclei. So this aberrance and the much larger size of these knockouts reveals that because endonuclease G has been removed, faithful replication and duplication and division of these parasites is no longer occurring. And finally, we wanted to test the infectivity of these null mutants. In Figure A, we can see that both knockout and wild-type parasites were phagocytized by macrophages. There's a much lesser burden of parasites when the knockouts were phagocytized. However, these knockouts still interact with the macrophages and still infect them, leading us to believe that in the host, they will still be able to interact with the immune system. In Figure B, we injected the ears of biopsy mice with both the wild-type and the knockout parasites. We found that when the L. mexicana wild-type were injected, 5 to 6 millimeter lesions were generated. However, 10 weeks post-infection, when the knockouts were injected, no lesions were present at all. This reveals that our knockout is actually safe and does not cause overt lesions in the mice. In Figure C and D, we measured the parasite burden in both the draining lymph nodes and the ears of the mice. We see a significant decrease in the parasite burden when the knockouts were injected into the mice. In the ears, we see a 7 log-fold difference between the number of parasites present. There's also controlled growth. So these parasites that are knocked out, that have knockout endo-G, can still interact and still live within the host. However, they do not cause any harm. So we have generated a novel Leishmania strain, the first ever wherein endonuclease G has been deleted. This attenuated L. mexicana strain is the first that is safe as a vaccine because it does not cause overt lesions. It has the ability to actually infect the macrophages, as seen by the previous experiment, and it has a lifespan long enough to provide robust immunity. It remains within the system for at least 8 weeks, allowing interaction to occur with the immune system. We had a patentability analysis done by a lawyer, and we found that in the past, only 2 L. mexicana knockouts have been made. One died within 8 hours, unable to interact with the macrophages or the immune system at all. The second could not even infect the macrophages, defeating the whole purpose of a vaccine. So in summary, we have generated a live attenuated parasite strain, the first ever that is patentable and has an excellent likelihood of being the first ever vaccine for cutaneous leishmaniasis. Thank you. And we'd like to acknowledge the Siemens Foundation, George Washington University, the FDA, and our mentor, Dr. Sreenivas Gannavaram, who helped us throughout our project. Thank you.